Hi, my name is Tatiana Belova and I'm working as a postdoc in the Center for Molecular Medicine in Oslo, Norway. Today I would like to present our work on heterogeneity in the gene regulatory landscape of leiomyosarcoma. Leiomyosarcoma is a type of cancer that can develop in soft tissues. It's a rare but aggressive cancer. Therefore, due to its rarity, it represents an understudied group of cancer. Characterizing intertumor heterogeneity is essential for selecting suitable cancer therapy. As it has been shown before, presence of diverse molecular subtypes of patients can be associated with disease outcome or response to treatment. Also, gene regulatory networks have been shown to be useful tools to study gene regulatory landscapes. These networks are represented by interactions between transcription factors or other regulators and their potential target genes. In our work, we perform a detailed analysis of interpatient heterogeneity in leiomyosarcoma. We do this by modeling large-scale gene regulatory networks using two methods, PANDA and LINUS. Then, to characterize intertumor regulatory heterogeneity, we propose a simple but efficient approach which we name Porcupine, which stands for Principal Components Analysis to obtain regulatory contributions using pathway-based interpretation of network estimates. Here is a pipeline of our analysis. We modeled individual patient gene regulatory networks for leiomyosarcoma patients from two datasets with PANDA and LINUS. PANDA produces an aggregate network by integrating information on protein-protein interactions between transcription factors, prior information on transcription factor DNA motif binding, and gene expression data. Then we applied a LINUS method to obtain patient-specific gene regulatory networks. And then we applied our new tool, Porcupine, to identify significant pathways that capture heterogeneity in gene regulation across each dataset. And at the end, we have compared results to obtain pathways that are significant in both datasets. So here I would like to present an overview of our method Porcupine. Porcupine takes as input individual gene regulatory networks and a GMT file with pathways. So these gene regulatory networks are represented by interactions between transcription factors or other regulators and their target genes, and these interactions have an estimated edge weight. Porcupine goes through following steps. First, transcription factor gene edge weight information is extracted from each individual gene regulatory network for all genes belonging to a certain pathway. Then, principal component analysis is performed on the pathway-associated transcription factor gene weight matrix. The variance explained by the first principal component is extracted. Then, the amount of variance explained by first principal component is compared to the expected amount of variance explained, which is obtained based on 1,000 randomly generated gene sets of the same size as the selected pathway. Effect size is calculated. These steps are repeated for each pathways, and then p-values obtained are corrected for multiple testing. So we applied Porcupine to two datasets. One is TCGA and another dataset is from the German Cancer Research Center. And we have identified 37 shared pathways that capture heterogeneity in each dataset. Among these pathways, we identify previously known pathways as well as pathways that have not been described before. Newly identified pathways could represent potential targets for treatments of subgroups of leiomyosarcoma patients, such as pathways involved in FGFR signaling and STL4 inhibitory signaling. Additionally, using Porcupine, we could also highlight genes and transcription factors that are important drivers of heterogeneity among leiomyosarcoma patients, including genes such as PP2, R1A, and RB1, and transcription factor E2F8. In summary, Porcupine allows to uncover patterns of interpatient heterogeneity at the level of transcriptional regulation in the cell and identify pathways that can potentially be targeted in the clinic. It thereby provides the first step towards implementing network-informed personalized medicine.